Hello, everyone, and welcome to the Meisam Express webinar series. Thank you for joining us. Today's topic is on CART gamma delta T cells generations using the CRISPR's AAV approach. So cell therapy has transformed cancer treatments, yet the current autologous CAR-T models remains costly, complex, and slow, placing it out of the reach for many patients. The gamma delta T cells offers compelling alternatives. They can be engineered as off-the-shelf allogeneic products with inner anti-tumor activities and slow risk and graft versus host diseases. By using CRISPR's AAV engineering platforms, today's sessions explore how next generation the CAR gamma delta T cells can overcome the key limitations of a traditional CAR-T cells, pair the potent preclinical efficacies with global scalabilities and advanced candidates such as CD38 CAR and then GD2 and nuclear CAR gamma delta T cells. Now it's my pleasure now to introduce our speaker today, Dr. Meisam Naeimi. And he's a research assistant professor of pediatrics at Ohio State University College of Medicine. He's also director of CRISPR's gene editing cord and then the principal investigators in the Centers of the Childhood Cancers at the Nationwide Children's Hospital. And Dr. Naeimi is a internationally recognized expert in gene-edited T and then the gamma delta T cell therapies for high-grade gliomas, TALL, and other hard-to-treat malignancies. His team has pioneered the CRISPR's AAV-based platforms to generate off-the-shelf the CAR-T gamma delta T cells, including the CT1838 and the GD2 and then the nuclear-directed construct. For today's sessions, Dr. Naeimi will speak about 40 to 45 minutes. Then we will open it up for a live Q&A. Please drop your questions into the Q&A panels in the Zoom chat at any times. We will collect them and then address them as many as we can at the end. Just to remind all of you that this webinar has been recorded. Now, without further ado, let's join me to welcome Dr. Meisam Naeimi. Okay, it's yours.
Hello, good morning, everyone. Where are you here in this globe? Here in the Ohio, it's morning, it's 10 a.m. And thank you, Meisam Express, for having me today. It's a pleasure to be here. I am Meisam, as Ian mentioned, that I'm an assistant professor of pediatrics here at the Ohio State University. Let me share my screen and start diving into what we prepared for you today. These are my disclosure. I'm a co-founder and chief scientific officer of a company I built called CARTx Therapeutics. I'm co-inventor of some technology presented here, and I received some payments from Sanofi and Chiatis. So overall, cancer immunotherapies basically is when you get a cancer in your body, a lot of immune cells start to migrate toward that cancer, start recognizing them and start killing them. And if you see here, they go through bloodstream into the and getting closer to the cancer. In the case of cytotoxic T cells, they need to be primed by antigen-presenting cells, recognizing the cancer tumor antigen, and cytotoxic T cells, they get close to the cancer and get activated, release perforin enzyme on other cytokines to kill the cancer cells. So basically, the cancer immunotherapy and cell therapies, you get those blood from the patients, get them into the cells, expand them to numbers, and put them back into the patients. And now you have cells, immune cells that can be either NK cells, helper T cells, gamma delta T cells. You have more in number, and then there are higher active, and they're more active in producing a lot of cytokines to kill cancer cells. So this is the basic of cancer immunotherapies using cellular therapies such as T cells, NK cells, or gamma delta T cells. But in the case of alpha beta CAR T cells that have been FDA approved, it's very promising. 90% almost of some of these patients treated with CD19 CAR T cells, they get response out of the treatment. But you just consider that there is a complexity exists for manufacturing, logistics, and also shipment of these cells. Just I'm gonna walk you through a little bit of these steps. You have a patient's bloodstream. You have to get the cells. This is called autologous CAR T cells. It means that you're getting it from the patients and you're isolating alpha beta T cells. You isolate the T cells from a hospital, and then you consider that you have to be in a state in the United States that have this something called apheresis to get your T cells out of your body. And then also, if you're not in the US or in the Europe, or in China or places that they have the capability of starting this material apheresis, you basically are out of options. Then you have to send the cells to manufacturing sites. Let's say it's a Kymriah or a Bluebird or whatever CAR T that you're prescribing to your patients. It has to be traveled across the globe to the manufacturing sites. And then complexity related to that, including cryopreservation, shipment, and then getting a very nice product into the manufacturing sites, it's challenging. Going back to the patient, your patient is heavily treated with chemotherapies. Their T cells are exhausted, they are tired. Your patient is as well. And sometimes they don't have even enough T cells to give you. And then you go back and then you add the virus in it that makes the CAR or chimeric antigen receptor now can recognize the cancer cells in a better way. And then you have to grow them more, pack them, cryopreserve them, put them on a plane, and then send it back to the hospital that the patient was admitted to. All these things that in autologous setting and all this complexity gonna cost $500,000 per patient. So you consider that you're in a country that you don't have access to all this stuff, and even you are $500,000 a dose, it's insane. It's very expensive. And consider for a second that your patient has a T cell malignancy. You have a TALL, T cell lymphomas, or any type of T cells that have been affected. So that patient is basically out of option for autologous CAR T cells because you can distinguish very well between the healthy T cells to do the manufacturing of your CAR T cells versus malignant T cells that you want to kill. Everything will gonna contaminate your product that you're going to ship it to the manufacturing site. So in my research and in my team, we've been always trying to find alternatives. And you cannot get alpha beta T cells from someone else. The reason is the host T cells or the CAR T cells will recognize the host immune cells or even the entire body as a foreign antigen, and they start attacking that. That's called graft, which is your CAR T cells, versus host, which is your patient and disease, and it can be lethal. So using alpha beta T cells non-edited from someone else has been really challenging. But there are ways to do that, and one of them is a CRISPR technology. In the CRISPR technology, you have a Cas protein, and also you have a single guide RNA that you combine this together, and it's called now Cas9 complex. And if you want to take blood from someone, a healthy donor, you send the CRISPR into it, and they're going to recognize the TCR or track locus, which can recognize the foreign antigen, unwind the DNA, bind exactly the complementary sites of the guide RNA DNA, and then activates the Cas protein, which is an endonuclease, so that it can make the DNA at the track locus cut. And now you have an opened up DNA that needs to be fixed and it needs to be repaired. And it's usually repaired in a very random way. So ATCG comes in and then puts in that place, and it puts your gene out of frame. That means it's turned off. Now you have a knockout that you can use a normal T cells from healthy patients to knock that gene out. And also you can do other stuff, like you can knock out PD-1, you can knock out whatever, and then you can put them back into the healthy donor. I'm sorry, a patient, and now you have an off-the-shelf allogeneic setting. But again, all these things you have to do. And if you are not familiar with CRISPR editing in a clinical setting, a single guide RNA that you purchase here is $500,000, and you can't grow T cells, alpha beta T cells. Just remember what I told here. You can't grow alpha beta T cells for so long because they get exhausted. And so you have to keep manufacturing more doses and more doses and more doses to get to serve enough patients around the globe. You can use like not only the conventional CRISPR, you can use base editing, that you can just change a single nucleotide to something that can introduce a premature stop codon and gene knockout, and now you have another alpha beta T cell allogeneic CAR T cells. And Dr. Kasim from UCL published this data a few years ago, and he treated 3 patients that this little kid is, or it's in remission after 3 years using someone else's CAR T cells. And that means that it works. Alpha beta T cells can be allogeneically used, but it's still, I mentioned about the cost of manufacturing, that can be break down to the number of doses that you use, but it's still, you can't grow them so much, and you have to always consider other complexity to the trial as well. The other thing is, most of the people, we mentioned about virus that you use to express the CAR at the surface of this alpha beta T cells, right? And these are randomly integrating viruses or vectors. So the CAR T immunotherapy outcomes correlate with the genomic modification by the vector. What does that mean? It means that if your CAR getting inserted in a place into the genome that you're not really want it to be, that affects your clinical outcome. Your T cells do not get activated very well. They don't proliferate very well. They don't stay in blood for so long, and so forth. So it is very important to understand that complexity related to using a viral vector, especially the lentiviral vectors, which are basically HIV vectors. It's been a little bit of data out there. It's a very, very, very low risk of a secondary malignancies to the primary malignancies in the T cells that you genetically engineered. It's pretty low, so we don't want to get into that, but it's been mentioned several times in the last few years. So what my lab has done, we thought that, okay, if alpha beta T cells are expensive to genetically modify them, to make them off the shelf, let's see what else we can find in the blood that they are, they have the capability to recognize cancer, but do not recognize the recipient. So it can be off the shelf or allogeneic setting. And then we can also genetically modify them, but not lentiviruses that they get insert themselves into the genome in a random fashion. We can make them in a site-directed insertion, and if we can immediately have them available, and that reduces the cost for 90%. And that's incredible, especially for those patients that they have T cell malignancies that have no options. I'll get more into the detail why CD38 and how it is important, but just consider that we are now wanted to start describing gamma delta T cells. So the gamma delta T cells are coming out of the progenitor T cells, out of hematopoietic stem cells, but in some time during the development, using some of the transcription factors, they can get selected to the gamma delta TCRs, and then they can get to different subtypes of gamma deltas versus the CD4 and CD8 alpha beta T cells, they come out a little bit differently. So it is important to know that the gamma delta T cells and alpha beta T cells, they come from the same progenitor cells and the same hematopoietic stem cells. But some type of biological effects make them to be different from alpha beta T cells. They've been discovered in 1984 to 1987. They could see that they've been expanded in HIV patients. They figured out they have roles in autoimmune diseases. They are having some roles in immune surveillance. And also they could have other they can form memory. This is extremely important in some of the diseases that we want to treat. You want to get your T cells to stay in blood longer and longer and longer as possible. And the first CAR was introduced in gamma delta T cells in 2004, and the development has been going on, and 2025 that we have some clinical trial running. If you remember from the first video, that alpha beta T cells, they need to be primed by antigen presenting cells in order to recognize the antigen on MHC1 and MHC2. So it means that they have to know what they're going to target in order to target. And that limits the alpha beta T cells, and that's why you do need for sure having a CAR on the cytotoxic T cells to be specific to what you want to target. In the gamma delta T cell activation, on the other hand, is MHC independent. They can recognize their targets by interaction between the ligands. It can be MIC-A, MIC-A, MIC-B, or B7H6 or 41BB. They do have a very specialized receptor for each of these ligand on their cell surface. That means that they can just directly go and recognize the cancer cells in a MHC independent manner. And that makes them very unique. It is very similar to what NK cells are. The NK cells also, they do the same thing, but the NK cells, they don't proliferate that much in response. I'm saying that much. There are very deep biology and knowledge in the NK cell field that I would be happy to discuss. But consider that there are similarities, there are differences in both NK and gamma and alpha beta T cells. There are some controversial comments about the anti-tumor effects and pro-tumor effects of gamma delta T cells. There's some data out there that in mouse, they've shown that some of these IL-17 producing gamma delta T cells can inhibit the CD8 positive cytotoxic T cells. Those kind of cells haven't been recognized very well and characterized very well in a human setting. Human and gamma delta T cells and mouse gamma delta T cells are quite different because their biology is different, the repertoire of the TCR are different, their antigens that they are engaging with, receptors that they engage with antigens are different. So you should kind of consider those biological differences between two species. One of the very famous papers that came out in 2015 in Nature Medicine, they looked at tumor-infiltrated immune cells across 39 malignancies, and both solid tumors and also other hematological malignancies. And the only immune cells across all this malignancy that showed to be favorable to the cancer outcome was gamma delta T cells. There are some technical consideration to this data that was because gamma delta T cells are pretty low in frequency in the blood. Sometimes they could only find 1 or 2 or 3 or 10, and they counted that in their analysis. But still, it was very significant across different tumors. Gamma delta T cells, as I mentioned, they're recognized by their their TCR. They have a repertoire of the TCR rearrangement, but the most important and more prevalent ones in the blood are VD2s. The VD2s, they can recognize BTN2A and BTN3A antigen on the cancer cells, and also they can bind to some of the antibodies, so you can combine them with antibody because they are CD16 positive. And as soon as they get engaged with this antigens, they can kind of configures their TCR on the surface, and they get activated. There are other type of gamma delta T cells, and they are non-VD2. They are VD1, 3, and so on. That they have more of other types of receptors, such as NKG2 D, that makes them very unique and makes them to reside in tissues instead of being in the periphery. During our time in our life, the frequency of gamma delta T cells subtypes change. It can be at the VD2 at the very beginning, and VD1 a little bit less, and then it changes at birth. So you have more of VD1 dominated at the birth, but then VD2 comes back, and then you get older, and it comes the VD2 goes down and in elderlies. That is very important because this is showing you that exactly which type of donor you want to use to get your gamma delta T cells from, which type of gamma delta T cells you need, and which age you have to go and find a healthy donor to give blood, and then to be your donor for your product. There have been data showing that this ratio between VD1 and VD2 changes in a healthy status versus the cancer patients that you see here. And also, when their peripheral circulation also can change, like if you are on the chronic stimulation versus you get the therapy, and that changes how this VD1 VD2 ratio changes in your blood, and it's pretty important. One of the clinical data that came out from the non-engineered gamma delta T cells was very promising from a company called Innate Bio, that they treated 10 patients with a non-edited. It's a haplo setting, so it's I don't want to get into the clinical part of it. I'm not the best person to describe it, but they got almost 100% of patients, they responded, and most of them now still in remission during the years and month after studies. So it's really, really, really promising. But the problem is there are very few gamma delta T cells in blood. It's like 0.5 to 5% of your PBMCs. So we need to get them out, and then we'd have to grow them. There are many ways to grow gamma delta T cells, but most of them, they are based on zoledronic, which is a biphosphonates molecules, and then they primarily grow VD1. They are MICA and IL-2, you get VD2, and then you get VD1. But what we like to do is mostly getting a polyclonal VD1, VD2, and VD1, VD2 negative population, because we believe that every single subtype of the gamma delta T cells, they have their own properties, and they need to be studied perfectly. And all this expansion protocol changes the way that they act. Their transcriptomic changes. If you look at the day 0 versus day 7, and then day 14 and day 21 post-expansion, they are quite different. And you see that differences also when you plot them, and then you compare 2 days. And it's important to understand those differences, how it can affect your overall growth of the cells, the properties of the cells, and how they can act when they see a cancer cells. In a genome editing point of view, also it is important. If you remember, I said if you want to use CRISPR, it makes a cut, and then there are enzymes that they stepped in to repair, and that's called non-homologous end joining. And then I said that they added some nucleotides and make a frame shift and premature stop codon and then stopped it, right? But if you want to use this cut to put in something, then you have to go through homology-directed repair signaling pathways. And the enzymes and proteins that they are involved in the homology-directed repair are different from non-homologous end joining. And this is particularly important, as I mentioned here, when you grow the cells, they change their transcriptomic. So we wanted to hit the cells and genetically engineer them when they have the highest amount of HDR in their cells activated. So we could put a CAR in a site-directed manner using homology-directed repair. But what we wanted to do, basically, we also use a site-off systems or a deep profiling to understand what we are targeting. And if you look at here, for example, this is an NK cell data. I have similar for gamma deltas, but this is important because it came from our lab and Dr. Dean Lee's lab. That you see here, like if you have not treated, you have a lot of live AML here. You treat them with the wild type NK cells, you get rid of a lot of these cells. But if you have a CAR NK, you get rid of a lot of them. But if you get closer, you have a small population here still surviving. And this is very important for us because they were CD38 positive. And also, if you look at the single cell RNA sequencing, also you can understand what survives and what is sensitive to your treatment. So that's what we use in our lab to look into sensitivity versus resistant mechanism in the cancer cells when you co-culture them with or treat them with the cancer with the treatment that you are interested to introduce. So going back, I said we need to grow the cells, right? And the way that we do it here, we do a short stimulation of our cells with antibody, which is pan gamma delta TCR for almost a week. And then we co-culture them with the feeder cells that they are 41BB and membrane-bound IL-21 expressing cells. They are K562s for almost 10 days. And then you see that they are all CD3 positive. They are not alpha beta TCR positive. That it shows the beauty of our system because you can't put a product into your patient that you have contamination, high contamination of alpha beta T cells. If you remember, I talked about the graft versus host disease. So basically, if you are not giving your patient a clean product and not contaminated with a lot of alpha beta TCR T cells, you get graft versus host disease and not acceptable to the FDA as well. And you see that we said we want a polyclonal system. So you see here we have VD1 presented, VD2 presented. They are all CD4 negative, and we have a population of CD8 positive cells here. And there are also some of them that are CD56 positive. And you see how clean they distributed. We genetically engineered them. I tell a little bit more about that, but then we can grow them to billions and billions and billions just by growing them and co-culturing them with feeder cells that we have. We have done this for 8 to 10 weeks, and each week we get about 100-fold changes. 100-fold changes means if you start with 20 million gamma delta T cells from a buffycoat, you ended up getting trillion cells, and that's enough for 100 to 1,000 doses based on what you want to put in your patient and the dose escalation studies. And that means that why and how we could decrease the cost 10 times less just by figuring out how we can isolate them, how to grow them efficiently, when to genetically modify them, when I said all those enzymes are up and the cells are ready to get engineered, and then regrow them. So it's basically that also decreases the cost of material that you have to use for gene editing because you edit very few cells and then keep growing, keep growing, keep growing. So you don't have to genetically engineer billion cells. You just engineer 40 millions and you get trillions after 3 weeks. So we started to target CD38, as I mentioned. And one reason was we started with NK cell program. We saw that multiple myelomas could be a good target for us. We use CD38. And if you target the CD38 with daratumumab or rituximab, NK cells, gamma delta T cells, through their CD16 and antibody-directed cell cytotoxicity, they get activated and they kill the CD38 positive cells. But there is a problem here that T cells and NK cells, they are also CD38 positive. So the daratumumab and rituximab, they bind to your ex vivo expanded immunotherapies. They get recognized by other cells that you put into the body, and they kill each other, and that's called fratricide. So we thought that now with this the protocol that we invented, we could genetically engineer the NK cells to get rid of CD38, so the antibody will be redirected to the cells that we wanted to do. But if you remember here, if you see here, we are using a combinational therapies of antibodies and our immunotherapies. So basically, this is expensive. And this is not so expensive, but when you have to keep giving your patient antibodies, it's getting expensive. So we thought that what if we make the CAR and we just use the antibody that we are using there and then genetically engineer them to be a CAR or chimeric antigen receptor, and then put them on the T cells or NK cells by site-directed insertion of our CAR into the CD38 gene. What does that mean is now we have cells that they are CD38 knockout because we used CRISPR to open up the DNA, and they are CAR positive because we are putting the CAR using these homology arms in the activated cells delivered by the AAV into the site that we opened up. So it will be a site-directed gene insertion. It's not like a lentiviral approach. It's not a random insertion. It's a site-directed insertion that now gives you the CD38 CAR, CD38 knockout cells. We made different type of this. We published it last year, and you see that how the cells grow very well. They can kill AML, multiple myeloma, TALL, and much more, because all this hematological malignancies, most of them, not all, sorry, that they are CD38 positive. That's why it's called CD38 is a pan-cancer targeting antigen, and why with a single production of this CD38 knockout CD38 CAR, we could target many different type of cancer, including the T cells malignancies. That if you remember at the very beginning, I mentioned about the T cell malignant patients, they are really having hard time if they relapse or get a refractory disease to get treated with autologous T cells. He said that we can't really distinguish very well between the healthy T cells versus the malignant T cells. So we went back to the optimization. We said that we isolate our gamma delta T cells and then co-culture them with the feeder cells, and then we keep growing them, electroporating them with the Cas9 RMP, transducing them with the AAV, and now we have a CD38 CAR and CD38 knockout cells. The efficiency, almost 100% knockout. So we have a 98% CD38 positive on the non-edited gamma delta T cells. After the knockout, you get 0%. And the CAR, we get almost to 60 to 70% CAR positive that we get here. And you see that there's no CD38 left in this donors. We mentioned about site-directed insertion. So we had to make sure that, first of all, after all the gene editing that we do, the chromosomes, they stay stable, because you cut the DNA. You don't want this piece of DNA from chromosome 10 goes and binds to somewhere else. Or if your CD38 is in chromosome 4, you don't want to reverse repair it. So you have to look at the integrity of your chromosomes using this technology that we use. And if you see here, we also looked at where exactly we put the CAR. And then you see this dots here, and it's exactly in chromosome 4, showing that we specifically inserted the CD38 CAR into the CD38 gene in a very site-directed manner, and there is no random insertion throughout the genome, which you see with the lentiviral vectors. We again tested these cells, and then you see how this is the no treatment JORCAT, which is a T cell malignancy cell line TALL, and then you treat them with the wild type and CAR. You get very good degrees of control. But I'll show you later. It is important to also always look at them in vivo. We got some samples from patients that they relapsed and they died. So we got those samples, and we saw that our CAR it's extremely effective against those patient-derived samples. A single injection with no cytokine support controlled the very aggressive TALL in mice. Guys, this is extremely important to consider, that a single injection, no cytokine support, with only 10 million of gamma delta T cells could control the TAL like this, is something that we should consider promising. And we thought that, okay, now our hematological malignancies and CD38 system works. What about solid tumor? And then you see here, these are the GBM or brain tumors. They're growing, and then small little things here. You see there are gamma delta T cells, and then you see how these cells grow versus the gamma delta T cell grow. This is a real-time IncuCyte data, so you can't fake it. You just look at the data and you see the differences, how they grow versus how they get controlled by our therapies. We tested these in neuroblastomas. We tested this against DIPGs, is a very aggressive brain tumor. Everyone does CD19, so we did the CD19 as well for B-cell malignancies and autoimmune disorders. And at the end, I wanted to thank Dr. Ashkar for introducing me to the world of gamma delta T cells. He's at McMaster University. We just published the VD1 CAR gamma delta T cell against ovarian cancer. A single injection controlled the tumor growth. With that, I want to thank everyone in my lab, in my team, Nationwide Children's Hospital, Cancer-Free Kids, and I want to give you more time to ask me questions rather than me talking. Thank you so much.
Thanks, Dr. Naeimi. Let's see, we have some questions from Jabe Cho. Great talk. I'm wondering if GDT cells do not get exhausted, unlike ABT cells. If not, why not?
So if you're asking about the growth, so in alpha beta TCR, to grow them, we use alpha beta TCR stimulation. In addition to CD28 co-stimulation that we add into the mixture, and that exhausts them because it's a constant stimulation. While here we did a short period of time of stimulation, and after that we got them going using our feeder cells by engaging them with the 4-1BB ligand and membrane-bound IL-21. So we did see that if we keep stimulating them with alpha beta TCR, they get exhausted. So we changed the approach. We said it's like a short period of stimulation and then changing into non-TCR stimulation, and that helped us to not letting the cells get exhausted.
Thank you. So we have another questions from Veldhit. can we use the CAR gamma delta cells to preclinical uh, genealogic oncology projects like ovarian tumors?
Potentially, yes. So you saw that we got some data from ovarian cancer, so I believe that would be the same, similar thing.
Great. Thank you. I have myself actually have some questions. I'm more familiar with the molecular biology. So do you think your the CRISPR systems with AAV is suitable for different looking different CAR systems, or there's a like limitation with the site specific insertions?
So we have made, I think, over 20 cars so far using the system. And the car has a size of 3 kb, and you include some regulatory elements to your car, and then you add some homology arms to it, and it still can almost fit any type of car that you want into your system. So we did not see from an engineering perspective any limitation of using AVs. And then consider using AV alone, it doesn't work in gamma delta T cells or NK cells. They need to get inserted. That's why we combine them with CRISPR technology to be site-directed insertion. And the other things I didn't mention here is NK cells and gamma delta T cells, their job is to attack viruses or to treat viral infection. So, and one of the viruses they get activated against is lentiviruses, which is basically HIV, right?
Yeah.
One reason that um, it works, but it's not as effective as our system is when you are transducing the cells with AAV compare versus the lentiviral vectors. So we have not had any technical engineering issues using AAVs in our systems.
Okay, great. And I just saw your the cell papers the with the natural killer cells and use the electroporations. And I also heard that there's some researchers in Sydney University, they have used the nanoparticles packet where they try to do the CAR-T engineering in vivo. Do you think do you see that your this way you put in the casts and then the donor together, maybe because I also heard that this LMP you can pack actually luciferase, maybe you can pack the enzymes for your system as well.
So let me get your question clear here, that you want to use in vivo CAR production, right? And you want to package all the elements that I use ex vivo to engineer the cells in vivo, correct?
Yes, maybe it's sort of opposite what you're proposing, because your approach is the selling point is that you can scale up because you can ex vivo engineered kind of thing, grow lots, and there's another approach is basically you don't have to get the T the immunocell out. You just use maybe nanoparticles, maybe decorated with say antibody that can target the specific cell type, and then
You consider for a second that your patient has a T cell malignancies. How your LMP and mRNA can distinguish between T cells that are healthy versus the T cells that are malignant.
yeah, that would relies on a very specific marker, but that's quite tricky. Maybe that's you have to do outside of the
Yes.
for cell sorting. Yeah.
The other thing, but it's doable for CD19, you can do it because you have, or BCMA, you have tons of clinical data that you could rely on. We were in a meeting, and one of the big names in the CAR-T field was telling us that, look, you want to do in vivo gene editing for manufacturing CAR-T, you can do it, but for those uh, applications that you have enough clinical data for, for those such as CD19, CD20, or BCMAs. If you want to go beyond that, you don't have enough safety data to put and generate and make your patient's CAR factory, right? What is coming after that? What are the side effects or off targets of your CAR? So I think it's doable, but it's very application dependent.
Great, thanks. It's, good to learn from this talk. Okay, we have another two questions actually from first one from Hodik. How is the ex vivo expansion of the GDT cells, as it is very slow percentage, very low percentage in peripheral blood? How many fold of expansion do you get in average after the CAR insertions?
So it's similar to our non-edited cells. The way that we optimize that is almost similar. We get 100 folds every week. And that means that in 3 weeks, we get thousands and thousands of folds every time we grow the cells. So the way that we optimize the system, that's not really a limiting factors in our production. And it is really not important that there are very few at the beginning. As you remember, I mentioned that we figured out a way that we can now stimulate that very few cells, like 500,000 or 1 million cells, to get to 10 millions, and that we can grow that 10 millions now with the feeder cells easily to 1,000 folds every time we expand them.
Okay. We have another question from anonymous attendees. Fascinating talk. Just for my understanding, does this technology means that the feeder cells are potentially the main limiting factors?
Not really, because I don't know what do you mean by limiting factors in which stage of production. If you want it at the very beginning, to do some research in your lab, we will be happy to share the feeder cells under some material transfer agreements. If you're mentioning about the clinical production, we have also some a way to collaborate on that. But if you are mentioning of remaining feeder cells in the final product, we haven't had that issues. Especially with our NK cell program, we had to show to the FDA that we can easily say that our product is clean of those feeder cells since they don't grow, they are irradiated, and your final product will not have detectable remaining growing and live feeder cells in the final product.
Thank you. We have one more question from Victorians. Which gene would you suggest to knock out in a GDT cells to generate the CRISPR's AAV and GD CAR T cells for treating the TALL, for example?
So as I mentioned, we knock out CD38, and then we use CD38 CAR to go after the CD38 positive cancer cells. This way, the gamma delta T cells, they don't kill each other because they are also CD38 positive. And also we will have a CD38 CAR that is a pan-cancer targeting cells. So that has been our gene of interest for TALL. For other cancers, it's very different. So you can have, for example, in some of our products, we use TGF-beta receptor 2 knockout, GD2 CAR. So we can make the cells resistant to very enriched TGF-beta microenvironments, right? So it depends on the setting, depends on the disease. We can open up almost any gene and put your CAR into the gamma delta T cells.
Great. We have actually one more question coming. Yes. First of all, Aisha's, can immunotherapy enhance the lipid metabolisms that alters the B cells checkpoints to program the tumor microenvironment?
Can immunotherapy enhanced by lipid metabolism that alters B cell checkpoints to reprogram the tumor microenvironment. I'm not sure I understood the question.
Maybe saying it's a bit strange. This okay, maybe we can skip this one. So another next question from the Dave Wood. So he asked, have you tried on the GBM?
Yes, so the glioblastomas we did try, and the data is fascinating. I showed you some videos, and those videos are GBM.
Right. Well, so we have a question from Amand. It's a great talk, and he said if he missed your point, was the allogeneic GD CAR T cells tried in the leukemias or lymphomas?
So the GD CAR T, gamma delta T cells, yes, we use them for CD38 against T cell malignancies, and also CD19 against the B cell leukemias and lymphomas. So yes, Ayman, and very nice to see you here. Yes, we did it, and I will be happy to share more data since we are moving toward the clinical trial for that specific product. And one single injection of our CD38 gamma delta CAR T cells was enough to control the very malignant T cell ALL.
Okay, the next question is asking if any risk to target other CD38 plus cells.
That was a very nice question. Yes. So the CD38 CAR gamma delta T cells basically targets any CD38 positive cells in your blood. So that's why the patient that they will receive the CD38 CAR gamma delta T cells, they have to go through a programmed hematopoietic stem cell transplant. So basically our therapy, I didn't want to get into the clinical specs of it. It's a bridge to transplant for those that they are eligible for a hematopoietic stem cell transplant. And the problem with the T cell malignancies or other, you really want to get rid of the cancer cells and get them to MRD negative as much as possible before doing the bone marrow transplant in this patient.
Right. The next question from the Hardik. Can you tell us about the persistence of CAR GDT cells in your preclinical in vivo models?
Yeah, so we did one injection, and after 30 days, still we could find a very good number of cells in peripheral blood. It is important to understand the allogeneic product in general. They may trigger some host anti-HLA activity, and you get your cells depleted. We have some way to go around that. But we have found these cells to be quite persistent based on what type of gamma delta T cells are you looking into.
And I think we'll have one more question from Aisha's. Ask your opinion. So can we find out the gene responsible for autoimmune diseases using CRISPR technology?
So it's a very broad question, and I'm not a autoimmune expert, but what I can tell you is for anything for discovery, you could use something called CRISPR screening that you can knock out basically 20,000 genes all at the same time in 20,000 cells, and then looking at what gene was responsible to develop some sort of autoimmune diseases in your patients. But again, I'm not a autoimmune expert, so I can't comment on that. But we have some CD19 CAR gamma delta T cells that we are developing for autoimmune disorders in particular, and that is not related to what type of autoimmune you have and what type of autoimmune mediated mutation you got in your patient. Anything that CD19 B cell that causes the autoimmunity can be targeted using, potentially can be targeted using our methodology and our product.
Thank you for your opinions, share your opinions. Yeah, okay. Thank you very much, Dr. Meisam Naeimi, for such an insightful presentation today. And thank you all, all the audience members for joining us for this webinar. There are more exciting MedCan Express webinars coming up very soon. Our next confirmed speaker is Dr. Brad Sleeps from Waters and Eliza's Hall Institute for Medical Research. He will be talking about the anti-malarial drug targets. So please stay in tune for that. At MedCan Express, we're committed to fostering scientific communications and accelerating the pace of scientific discoveries. We thank you again for joining us today, and stay connected. And I look forward to seeing you all very soon. So goodbye for now. Thank you. Bye.
Bye-bye.